In this experiment, we studied particle motion and we tracked the movement of silica beads on a slide, and we watched and analyzed the movement under a microscope. We came up with an experiment which tested two variables, the size of the beads and the solution's viscosity and how it affected movement. We saw if these variables slowed down or sped up the particles and if the overall pattern of the movement changed. Our hypothesis was that the larger the size of the molecules, the faster that they diffused and they settled in solution because the larger particles have a greater momentum due to the momentum equation and thus are not shifted as much from their course by collisions with other particles. We also hypothesized that the more viscous the solution or the thicker the solution, the slower the particles would move because it would be more difficult for them to travel through a thicker solution. This means that we thought the molecules would move faster in water because water has a viscosity of 8.9 times 10 to the negative fourth pascals, and glycerol has a viscosity of 1.412 pascals, which means that glycerol is more viscous and thicker.
First, we set up and calibrated the microscope in the computer software. Then for each sample, we shook it up and used a pipette to place about a dime-sized amount on a clean slide. And then we took videos on the microscope showing the motion of the particles, and we did this multiple times to ensure the data was accurate. We then used tracker software to collect position data for a single particle in each sample, and then we made graphs from that data on Excel and analyzed them to determine particle motion.
This clip from our experiment is when we used the 5-micron silica beads in water. This video footage is from the microscope and shows beads settling in solution. At first the beads move very quickly because they are excited, but then at the end they are starting to slow down. This is the position graph for the 5-micron bead in water. In all the position graphs, the line of best fit shows the average slope, which is indicative of the velocity graph. This histogram shows the distributions of displacement for the 5-micron beads in water. This graph shows the position of the 5-micron bead in glycerol. Notice how the graph shows the beads settling at first and then stopping. This histogram shows the distributions of displacement for the 5-micron beads in glycerol.
This graph shows the 1.5 micrometer bead in water. Notice how it keeps moving for the entire time. These histograms show the distribution of displacements for the 1.5 micron bead in water. This is a position versus time graph for the 1.5 micrometer bead in glycerol. These histograms show the distribution of displacements for the 1.5 micron bead in glycerol.
So in conclusion, our hypothesis about the size of the particles was proved. In glycerol, the 5-micron beads were essentially settled and stopped by 5 seconds, but the 1.5 micron beads were constantly changing throughout the tracking period in the x direction. The same results occurred in water. The 5-micron beads settled very quickly, but the 1.5 micron beads consistently moved. Our hypothesis about viscosity was unfortunately not proven to be correct. We did not see any significant differences in movement in water versus glycerol. Therefore, the viscosity did not have as big of an impact on the movement of the particles as we initially thought. Overall, the energy is conserved within the system, but the system is not limited to only the silica beads, but rather includes the beads and the solution together. This means that the beads transfer energy to the solution through friction, which is why the beads stop, but is why the energy isn't lost.
So the motion of the beads in our solution differed based on how big the beads were and which solution they were in, like water versus glycerol. And we saw the beads move more quickly at the beginning of the video than as the video progressed because we had just shaken them up before we put them on the slide. We also noticed that motion happened in both the horizontal and vertical directions, so X, Y, and Z all experienced some changes. We noticed this because the beads would sort of fall into focus on the microscope and come from the top into settling near the bottom where we had the microscope focused. In the pre-lab, we expected the beads to eventually slow down and stop because it's not a frictionless environment and they're colliding with one another, and that matched our observations of the beads in the solution. In the simulation, the particles never stopped moving because friction and energy loss were not accounted for, but in our videos, there was not a frictionless environment, so it's expected they would diffuse and eventually stop. But they were similar in the fact that both the simulation and our real-life videos modeled change in momentum with collisions.